In the last session, we talked about how we can identify our staph unknowns. Now we're going to talk about how we can identify our strep unknowns. So remember, we differentiate our staph and strep based on that catalase test. So all of the tests that I'm going to be talking about next have to do with organisms that tested catalase negative. And they are going to fall on the strep side. So all of these flowcharts that I'm showing you are part of the lab that I'm going to be including in the folder for you. Again, go back and read the protocols for doing each of these tests so that you know what happens in the lab and also what each of these tests is looking for. So these are the tests that we're going to be performing on our catalase negative organisms, and these are going to be our strep species. So group A strep, group B strep, all of those organisms that I talked about on that side of the flow chart. So the first test that we would actually perform is to set up our hemolysis plates. For this test, we would be using blood agar plates. So I'm going to underline that right here. Blood agar plates are basically tryptic soy agar plates, but they're infused with 5% sheep's blood. The reason that they're infused with that sheep's blood is that if those organisms produce certain types of hemolysins, we're going to see different types of hemolysis on the plate. And hemolysis is when the bacteria breaks down the hemoglobin in those red blood cells. Depending on what type of hemolysis it is, we're going to see different results. So we would do an isolation streak. And if you remember in our isolation streak, our quadrant 1 has the most growth. And then as we go to quadrant 4, we're going to have less and less and less. One of the things that we want to do to help to differentiate between each of these strep species is to add an antibiotic disc. Now, these are discs similar to what we used in our antibiotic testing lab and looking for that zone of inhibition. So the two antibiotics that we use are bacitracin, and that disc has a B on it. The other antibiotic that we use is Opticin, and that has a P. The reason that it has a P is that it's also known as Taxo-P. So these are the two antibiotics that we would place in quadrant 1. The reason that we're placing them in quadrant 1 is that we have the most growth. So it would allow us to see that zone of inhibition, or if we did not get a zone of inhibition. So the different types of hemolysis that we would need to identify is number 1, beta, and beta hemolysis is complete hemolysis, so that's going to be complete destruction of those red blood cells. And if you look at the plate that has the B on it, it almost looks transparent. So the blood agar plates are very opaque. If you held them up to the light, you would not be able to see through them. But if an organism is plated that exhibits beta hemolysis, if you held it up to the light, it would look very yellow and transparent, so you could actually see right through it. Alpha hemolysis is partial hemolysis. So this is partial destruction of the red blood cells. And so if you look at the plate that has an A on it, if you held it up to the light, you wouldn't necessarily see right through it, but you might see a greenish hue. Again, that greenish hue is going to be from that partial breakdown and the hemoglobin starting to break down and exhibiting some of those different colors. So alpha has a greenish hue around it. Gamma hemolysis, this is lack of hemolysis, so no destruction of those red blood cells. And if you look at the plate with the G on it, you can see where that plate looks very opaque, so it looks that red color, so that is that blood agar. And then it has these little white colonies on there, and that's lack of hemolysis. So the key to identifying our strep species is to correctly identify the type of hemolysis. whether it's beta, alpha, or gamma. The reason for that is that it's going to allow us to go basically down a rabbit hole where we're going to be looking at different tests or looking at different antibiotics to help us identify that bacteria. So here's our strep flow chart. So the key to